So I'm a clinical molecular geneticist, so I work right at the interface of our clinical team and our basic research team. My lab is very focused on deriving cancer genome information from very hard to work with clinical material, biopsies, surgical specimens. So my talk is really going to be around the approaches to using next generation sequencing to basically read out somatic changes in the cancer genome. My only disclosure is that BI has funded a fellow in my group to do circulating tumor DNA analysis, which I'll touch on right at the end. So we're going to learn about these various things. It's a little bit different than what's in your book, but really try to become familiar with what is cancer genome variation, how can we read it out, and what are the strengths and weaknesses of those different approaches. Really the challenges of these beautiful figures you see in research papers and the challenge of moving that into real clinical practice, which is really my day job. And number 4 is, it's one thing to read out a list of variants. How do you actually put these into clinical practice? And I'll touch on this right at the end, especially around thinking how do we write molecular reports that clinicians can pick up and act upon. So I'd like to start with just what is the human genome. So this is one of my blood cells dropped onto a slide and stained. So this is a collection of chromosomes. And historically, it was literally a cut and paste, like paper and glue exercise to make a paste up like this. So here are all my chromosomes lined up, 46XY. And I show this just as a contrast to what a cancer genome looks like, where almost anything that can go wrong does go wrong. There are lots of additional copies, there are missing parts, there are rearranged pieces. And that's really the point of this slide is this is the exact same information, but now organized as a circle. So all the chromosomes are now as a loop around the outside. And all the colors are all potential changes in that genome. And the list there, question coming, is basically a list of what are the ways that cancer genome can be modified. Point mutations, copy number alterations, structural alterations, where a piece of one chromosome is now rearranged and stuck to another chromosome, as Jean-Yves showed. All these map to pathways and homologous regions, and layered on top of all of this are epigenetic modifications that aren't read out very well by traditional next-generation sequencing-based methods, but now there are really great tools to modify those protocols to read out methylated bases as well. So I put a grossly simplified next-generation sequencing-based approach. Now, what is it? There are a host of papers that will focus on all of these aspects, but at the end of the day, the goal is to really go from genomic DNA extracted from populations of cells, modify them to make a library, which is really just DNA that's compatible with a next-generation sequencer. There are lots of devices that do step 3 here, so as long as you have your DNA compatible with those sequencers, they're ready to go. And then computational analysis, which is a whole research field just on its own. So I'm going to touch on all these areas rather briefly, especially on the computational analysis. I'll have little words in red. These are bioinformatic tools that I use in my group, so people who want to do a little more reading or dig into specific tools, that's what the red letters mean. So this is literally what comes off of a next-generation sequencer. It's a massive text file of ACs, Ts, and Gs. So this is 25 reads of the 600 million reads that you'll get from one lane. So we actually generate 8 lanes of these. So you can just, by eye, appreciate the scale of data. So data generation is really not the challenge here. The challenge is really putting this into a meaningful order and into an order in which you can start to make comparisons across various reads and then make molecular inferences about them. So the first step in computational analysis is to take all these reads and compare them to a reference, and that's what this looks like. It's far cleaner. The way to read this slide is, this is a piece of software called the Integrative Genomics Viewer. It's free online from the Broad Institute, and all it does is take your human genome reference sequence, which you can also just download off the internet, and then it just aligns all the reads up to that reference. You can see all these reads look more or less the same. They all match perfectly to the reference, and I've intentionally chosen a region of the genome that doesn't have any alteration, so all of these reads are exactly the same. So you have hundreds of X, in some cases thousands of X redundant measurement of the exact same base. In this case, there's not a single change. This is what my original genome looked like. No changes, at least in this region. The goal of computational analysis is to look for differences. So this is just a cartoon of the exact same data I just showed you. So here's the human genome reference along the top. Here are all those little gray reads I showed. And then computationally, we're using these methods down here on the bottom to look for all these different changes that I talked about on the Murphy's Law slide. Point mutations, just a single base pair change in that read stack I just showed you. Indels, missing pieces of DNA. Homozygous deletions, these are just 2 copies of DNA completely missing, we infer this as the lack of data because there's nothing there to sequence. Hemizygous deletion, so one of your 2 copies is deleted. This is half as much data as you'd expect. And then copy number gains, more data than you'd expect in that exact same region. Translocations as well, where a piece of chromosome 1 is stuck to chromosome 5. We can actually infer that if part of your DNA sequencing read maps to one chromosome, the other part maps to another. And then all the DNA that doesn't match the human had to come from somewhere. Often they're actually pathogenic sequences. You can read out HPV, EBV, cancer-associated viruses all in the same DNA sequencing reaction. So this is, again, what real data looks like. This is also a question coming. The upfront laboratory protocol really dictates where your sequencing reads lie. So really, this selection of the genomic protocol right up front will actually dictate what you can detect. So here's what whole genome sequencing looks like. An entire region, just a sea of data, nice even coverage of reads across everywhere in the genome. Contrast that to whole exome sequencing, where really your reads are only piling up in regions that you can think you can interpret. So here's a gene down here along the bottom. You can see all the exome data is only mapping to protein coding regions. These are regions that are most readily interpreted. But you're missing all this regulatory variation. All the intronic and intergenic regulatory variants are missed by exome sequencing. And RNA sequencing is really letting the cell do the selection for you, where DNA is read into RNA, introns are then spliced out, and therefore all your reads, again, only map to exons. The advantage of RNA sequencing is it's quantitative, so if a gene is expressed at a high level, you get many, many reads. If a gene is not expressed, you get 0 reads, and all the other genes in between. The other benefit is you can do exon discovery. So while an exon is not described here, there are reads in this region. So you can actually discover new exons, new isoforms using RNA sequencing by letting the cell really do that exon selection. The other big benefit, certainly for clinical application of exome sequencing by focusing specifically on coding regions, is the ability to count mutations. This has become extraordinarily popular now with the advent of immunotherapy, where high mutation rate cancers like melanoma, lung adenocarcinoma are associated with response to immunotherapies. However, we have seen responses in some of these low mutation rate tumors as well. The other reason I put this out is there are exceptions to every rule. So there are high mutation rate pediatric cancers. Certainly in ovarian, there's a class of high mutation rate ovarian cancer. And this melanoma here is actually very, very low mutation rate, in fact, more in line with pediatric cancer. This is actually the only melanoma that was not sun exposed in this TCGA study. This was on the bottom of the patient's foot, no sun exposure at all, just very unlucky, got a mutation in an oncogene and developed melanoma that looked a lot like the other ones, but very, very low mutation rate. Just really illustrating that there are multiple paths to develop the exact same cancer. Some tumor genomes are highly rearranged, some have no rearrangements, and some are focused, and it's very difficult to say this cancer is a high mutation rate cancer or a low mutation rate cancer or a high rearranged cancer or a low rearranged cancer. In this case, lung adenocarcinoma, there's 3 examples, low, medium, and extraordinarily high number of rearrangements. Neuroblastoma, pediatric cancer, very, very low rearrangement rate, apart from this report of chromothripsis. In this case, highly, highly focal rearrangements of a single chromosome. I mentioned earlier methylation specifically. Epigenetic modifications that can regulate gene expression can also be read out by a modification to your genome sequencing protocol, in this case bisulfite sequencing. This is actually the output from a Lynch superpanel where we'll actually capture DNA from 5 Lynch syndrome associated genes, treat that DNA with bisulfite, and the effect of that treatment is really a laboratory modification of all the cytidines, of C's to T's, in only the unmethylated regions. In this case, the unmethylated promoter in normal tissue turns blue, so really just by eye, you don't need bioinformatics method to really read these out. Contrast that to an endometrial carcinoma, where all those C's were protected by the methyl group. They don't get changed by bisulfite, and really in this case, this is an MLH1. This is a methylated MLH1 promoter. So really using next generation sequencing data to read out yet another source of cancer genome variation. And at the end of the day, really all these data types are confirmatory and complementary. So really there's not a one-stop shop to read out variation in the cancer genome. So I've really listed the 4 different types of assays that I've described today. Sequence mutations, really something you can read out since we're doing sequencing on all of these platforms. Copy number by looking at changes in coverage, again, well suited to exome and genome sequencing, as well as methyl-seq. RNA-seq really much more suited to gene expression levels, rearrangements, pathogens. Really, RNA-seq is really the only way to directly read out gene expression patterns, although these may be inferred indirectly by looking at methylation alterations. Okay, so with that as context, sources of cancer genome variation being read out by next-generation sequencing, this has been done literally thousands, almost tens of thousands of times in ideal, often surgical, high-purity specimens. So this is a plot of the number of tumors analyzed by a very large consortium called the International Cancer Genome Consortium. So this includes the Cancer Genome Atlas, which is the American wing of this larger international umbrella organization. Each color is a different cancer type, and you can really see how just genomic data has piled up over time. So as of November, nearly 15,000 donors have had, across multiple projects, have had, just an example, millions of mutations detected. So really reading out mutations in ideal specimens is really no longer really a challenge. The real challenge is when you move into the real world of clinical biopsies and surgical specimens that aren't 80% plus tumor content. The first challenge, especially you look moving from research to clinical samples, is this use of formalin-fixed paraffin-embedded material. So this is actually a figure from my very first paper ever. This is still a problem. These are all DNA from lung adenocarcinomas. You can just see just by eye the spectrum of DNA degradation. You can actually, these are all done from the same hospital. These are all from the BC Cancer Agency. High molecular weight DNA, totally degraded DNA, and all forms in between. There was no real association with age. This absolutely destroyed case was a decalcified bone metastasis that actually had a very clear distinction. But otherwise, this was a real challenge up front. It's just how do we make use of highly degraded DNA? The other challenge is that tumor tissues are a mix of cancer and non-cancer cells. This is a metastasis to a lymph node. I've circled out the tumor cells, but you can just see by eye here, this is actually a pretty good specimen. The tumor content here is really only 50%. So as we're doing next-generation sequencing, half of our reads are coming from normal cells. We're just going to have to sequence that much deeper to find tumor-associated mutations. And the reason this is important is that cancer subclones, so populations even within the cancer cells, may respond differently to therapy. And this is actually a nice example in leukemia, where you start with the progenitor cell, it acquires additional mutations, and then within that cell population, there are actually subclonal specific mutations. So this cell has an orange mutation, this cell has a yellow mutation that's shared with this other clone. Here's this purple clone that has mutations totally private to itself that actually make up the bulk of the tumor cell population. Treatment with chemotherapy here actually kills off a large number of the subclones. So this purple clone actually happened to be quite susceptible to chemotherapy. But really, you do have this very small, minor subclonal population that then recurs, acquires additional mutations, and eventually results in relapse. These are the types of patterns we can read out to the base by next-generation sequencing, assuming that the assay run at this step here is done to sufficient depth to account for these differences in tumor content and subclonal patterns. So deep DNA sequencing really does enable readout of all of those sources of cancer genome variations. This is actually data from an endometrial carcinoma we've sequenced in collaboration with Amit Oza, really just illustrating this very flat copy number space, as Gabe alluded to earlier. There is a very, very small focal alteration here that actually has a point mutation inside of it. So really looking at these 2 hits informatically, very easy to pick these out. Modeling various combinations of genome duplication, also described as ploidy. So in this case, the ploidy, the number of copies of every chromosome is 2, just like a normal cell. But also the tumor content, in this case, being very high. We can be able to not just rely on a pathology review, but also being able to infer this informatically and making sure that those 2 data points are in sync. And then finally getting into what is the subclonal structure of this tumor. So in this case, each little tiny slice here, this is actually a hypermutated case. Each little slice here are are individual mutations. So here's PIK3CA. This is in this superclone. Here's a KRAS mutation, potential resistance variant, also in the superclone. A little collection of mutations that are at roughly 75%, another cluster of mutations that are only seen in 25% of cells, and subclones of subclones and subclones of subclones. Really enormous genetic diversity, but something that actually be summarized and interpreted relatively easily, even just by eye, assuming the right tools are read out with the clinical and the pathology information alongside. Moving to an extreme, the ultimate complex tissue is really circulating tumor DNA, and this is really the concept of using a blood draw rather than as an indirect measure of tumor-derived DNA rather than the blood itself, or rather than the tumor itself. The real challenge here is most of the DNA that you will find circulating in blood is actually from healthy or inflamed tissue, and is very, very, very minor contributor. The tumor actually is a very, very minor contributor of DNA circulating in blood, depending on the cancer type. Certainly the heme malignancies have a much higher tumor burden. I've seen reported as high as 40%. Solid tumors we've seen more around sub 1% concentration. So really sort of the extreme, the worst possible tissue really is blood itself, because almost every piece of DNA is from a normal cell. That's not to say that we can't find those variants. Coverage is money, so if you spend enough money or use a very targeted assay, instead of doing a whole exome or whole genome, focus it on relatively few genes, we're actually able to read out cancer genome variation at very, very low levels. And actually, just the sheer quantity of circulating tumor DNA actually mirrors tumor burden really quite closely. So this is a figure from a Stanford group, really showing that the volume of the tumor as measured by imaging in blue is really closely tracked by the specific fusion that's read out in circulating tumor DNA. So as the tumor shrinks, you see less circulating tumor DNA. As there's relapse or tumor growth, you see a corresponding growth or increase in concentration of ctDNA, all the way down in this case to no detectable tumor by imaging, or very little detectable tumor by imaging, and very little, if not zero, circulating tumor DNA in the patient as well. So this is very nice just by looking at quantity of circulating tumor DNA. The actual sequence is very useful here as well. So here's an example from my group where we ran a very small 5-gene panel on multiple myeloma patients, both their matched bone marrow aspirate as well as their cell-free DNA. And what we saw in this case, there were actually 4 mutations in bone marrow, so 2 KRAS mutations, both at very different frequencies, a BRAF mutation and an EGFR mutation. All 4 of these are all at different mutant allele fractions, but you can really see in blood, very, very low levels, but still a very nice relative relationship recapitulating the clonal structure in the primary tissue as well. And actually, here's a second patient where there were 2 hotspot mutations that actually resulted in the same protein change, but you can really see they're mutually exclusive. They're either on 2 different alleles or more likely 2 different subclones, a very minor subclone and obviously a very major subclone. But really using DNA sequencing as a quantitative readout of somatic variation, even in blood. So this is all great, measuring and quantifying cancer genome variation, but in terms of clinical action, the question for an N of 1 patient is, what are the targets in my cancer and what can be done about them? Certainly we can read out sequence structure, function. We can detect viruses, bacteria as well, Fusobacterium and colorectal being the most famous. And this is really the challenge, especially in clinical molecular genetics labs, that interface between the basic researcher, clinical labs, and clinicians who are really going to have to act upon this. And this is, I actually believe this is the real challenge in clinical genomics today. How do you integrate and communicate all this data at once? Currently, how do we report and share these results? This is really black text on white paper. It's really not scaling very well as we move to thousands of genes, multiple sources of cancer genome variation. This is the case, 3 mutations. This is a whole block of text just to describe one of those 3 mutations. As we get to hundreds or thousands of mutations, no one's going to want to wade through hundreds or thousands of paragraphs. And one potential revolution in this area is really trying to use web-based technologies, data sharing, something a little more interactive as a clinical readout, and really a place where we can start to integrate molecular and clinical data all in the same place. So this is CBioPortal. This is really an aggregator of cancer genome data. This is actually a portal we've deployed internally at Princess Margaret Cancer Center. The way to read this is actually a dashboard from a single case, from a single patient. These are months here along the top, and each little circle, numbered circle, is a different tumor. So in this case, we actually, this is actually done at Sloan Kettering. These are 4 tumors that they analyzed, 2 at the beginning, multiple clinical interventions. By the time you get to tumor 3, this clinical intervention continued until tumor 4, and then a final treatment here at the end. And really just by eye, starting to drill into how are these tumors the same or different over time. You can see hyperamplification in one point, really not seen in any of the others. Very common deletions seen across all 4 tumors. And this list just goes off the page, just hundreds of mutations. In this case, I've just picked out 3 that are shared across all 4. But really trying to reinvent, certainly for research, potentially also for clinical testing as well. What does that tumor look like at a snapshot in time, and how is it changing over time in the context of different treatments and potentially different trials, as patients will actually enter into multiple serial trials over time. So I'll leave it there. I had references on most of the slides, but here they all are here. Here are people in my lab and my funding sources. So I'm happy to take any questions at this time.